today. With that, we will start the recording. I will hand over to Claire, who will go through her presentation now. I can see the recording has started. So over to you, Claire.
Okay, thank you. Thank you, Philip, for the introduction. I will just do this. Please stop me if you don't see my screen.
Yeah, we're good.
Yes, okay. So thank you again for the introduction. So I will present today the work we performed here at Quality Assistance on multiple attribute monitoring in QC, alternative to the bottom-up approach. So let me start by saying a few words about Quality Assistance. We are a contract research organization. We are a private company with more than 40 years of experience. We are currently 250 employees, and we provide analytical services for pharma and biopharma companies. And we work in compliance with the current regulations. We work on all types of molecules from discovery to market, from monoclonal antibodies and ADCs to viral vectors, mRNA. You can see the list on this slide. And we are located in Belgium, in the French-speaking part, south part of Belgium, and we have all our labs on one site, which is one of our strengths, which makes things compliance samples. So let's go to multiple attribute monitoring. The concept of MAM, multiple attribute monitoring, was initiated in the US by Amgen. And here you can see on the screen two papers describing the concept. This is actually the one from 2015. It's the first one describing the approach. And the idea of this multiple attribute monitoring is to replace multiple liquid chromatography and capillary electrophoresis method by a single method. So commonly, currently, we would have, for instance, chart variant analysis and also clipping and fragment analysis performed by capillary electrophoresis, while the glycan mapping, the identity test, or the oxidation would be done by a liquid chromatography, but all in independent runs, while we could have a single LC-MS method to analyze all these attributes. So the approach is a bit more accepted in the US than in Europe. I believe there is more lobbying on the other side of the ocean. And also it's more established across industry in a non-GMP environment for product and process characterization. So the paper here on the right is one example of an application for product characterization. And so if we have a scheme here of what is multiple attribute monitoring, this is a scheme I borrowed from the paper cited here, which is a paper from the FDA. So there are two steps. First, method development, where we have a monoclonal antibody digested commonly by trypsin to have specific peptides. And in a method development step, the peptides are analyzed by LC-MS, which allows a complete identification of all the peptides, and it allows to determine which peptides are considered as critical quality attributes. Then in a routine setup, we would do the same sample preparation with the trypsin digestion, but the samples would only be analyzed by LC-MS. And with a targeted identification on the peptides which have been defined as critical quality attributes. In parallel to the targeted approach and the quantification, we could also do a new peak detection. To implement such method in a QC, there would be a number of challenges. The first one is that it is assumed to be complex because it requires MS data and probably MS trained users. To have a use in QC, we need to make sure of a number of things. We need to make sure that the performance, we need to check the performance of the method, which means checking the repeatability and reproducibility. To work in a GMP environment, we always take care of data integrity, and this means also working with validated software, so software which are compliant. And we always need to validate the method. And finally, one challenge would that the approach should be accepted by QC lab and authorities. In parallel to this, there is a big advantage that it would provide quite a number of data with a unique analysis. So we would be able to monitor and quantify critical quality attributes. There is the possibility to do new peak detection. And also because MS data are acquired, there is always a possibility to go back to previous data once a new attribute would arise, for instance. And because it's a unique analysis, I think it's time and cost efficient. So commonly, the multiple attribute monitoring is done on the peptide level, but today I want to discuss the middle-up approach. So this has already been reported. There are two examples here. On the left side, the paper from 2018, where they do this type of analysis for glycan profiling. And also because they have co-formulation of antibodies, they look at the ratio of the two antibodies. And on the right one, the paper from 2021, this is with an ADC. And there, the main aim of the method is to confirm and monitor the conjugation of the antibody with the drug. So in our work, the aim was to develop a MAM approach for monoclonal antibody at the subunit level. So we want a method that can be used in GMP environments with an automated process with output on CQA and new peak detection and implement SSTs. So let's go to see the results obtained on this topic. First, to work on the subunit analysis on the subunit level of antibodies, we've used the Genovis enzyme called Fabricator. It's an IDS enzyme. It's really specific. It cleaves only at one cleavage site below the hinge region. And it's the digestion only takes 30 minutes to 1 hour. So this is also quite fast. And so with this IDS enzyme, with the Fabricator, we obtain two species, the Fab region, which is roughly 95 kilodalton, and the FC region, which is 25 kilodalton, yes. And if we would use a reduction agent like DTT, for instance, then we cleave all the disulfide bridges, and we obtain three species of roughly 25 kilodalton, so the LC, the FD, and the FC subunits. So this is our experimental setup for the subunit analysis. So we use the Fabricator enzyme, as just explained, and followed by a reduction step with DTT. We used common reverse phase condition with a bioresolve column from waters, and we've performed this analysis with a bioaccord, so the RDA from waters as well. We acquire and process the data with Unify, and it's a fully compliant software. Sorry. And here is an example of a chromatogram obtained for the antibody adalimumab. So the UV chromatogram and the MS at the bottom. And you see the FC, LC, and FD peak. So we have obtained the three subunit species we expected. And here you can already see before, in front of the FC peak, a small peak, which is the FC, which is oxidated. So we will see this again later on. So we are able to separate this. The design of our automated process. First, we use the NIST mAb as an SST because it's a defined and known antibody. And we set a criteria on the UPLC level, on the retention time shift, and on the percentage UV area with multiple injection of this compound. Then we have criteria on the MS, and we check the percentage of modified light chain, and the percentage of modified FD. And all these calculations are based on custom fields in the software. And the second part of the process is for the sample, of course, for quantification of the defined CQA with the possibility to do the new peak detection. And we have the possibility to include alerts and limit on oxidation or glycan values, for instance. Before going any further, we check the performance of the method, first on the chromatography level by injecting several times, so 9 times the same antibody, but actually it's multiple preparation and multiple injection days. So on the chromatography level, we check the repeatability on the retention time and on the percentage UV area. And as you can see here, there is barely any error. So this was good results. Then on the MS data, we check the identification level. So again, with these 9 injections, and we see that the repeatability is here as well, good with the same species identified. Only for one of the compounds, there is a bit more error, but still we believe this is fine. It's still okay. And we also check the influence of ejected amounts on the MS responses by injecting the sample at different levels and check the MS response after deconvolution. And here as well, we have limited error bars. You can see them. It's quite minor, except for, again, one a bit more odd identification. But here as well, it's a small amount, and we believe this is acceptable. So to check, to make use of the method, we prepared a small case study. For this, we prepared oxidized samples. So we use a third butyl hydroxyperoxide for oxidation. And this oxidized sample, a regular control sample, and the NIST samples were digested, like we discussed before, with a fabricator enzyme and reduced with DDT. Then we made a mix of these oxidized samples with a non-treated sample at different ratios. So 0, 25, 50, 75, and 100%. And we made 3 independent preparations. All these samples were injected, so the NIST as our system check for the SSTs, and the samples to obtain results on the CQA and UPIC detection. And for all this, we have an automatic report of the data. So if we go through the data we obtain, first on the NIST, which is our system check, this is what the chromatogram would look like. So FC, LC, and FD, the 3 subunits we expect are present. And this was injected 3 times, I believe. And we have calculated error on the UV area and on the UV retention time. These values were good. We have set threshold for this based on the performance of the method. So this was acceptable. We checked the percentage glycation on the light chain and the percentage pyroglutamic acid on the FD. And here as well, we can set limits and thresholds because these values are reported, so we know what to expect. And this is a good way to check that the MS data makes sense. So as this is satisfactory, we can move on to the process of the sample. And here we obtain a list of identified subunits with their modifications. So this is a small example how it looks like. And we can calculate the relative percentage of each species at the subunit level. And in the software, it's easy to make a color code for values which would be above what is expected. So this is what we've done here. Of course, this is just an example. So the values are arbitrary, but we would immediately see that the sample is out of range and is not acceptable because there would be too much oxidation in this example. So in summary, we have an easy and fast method with good reproducibility, which would be adaptable to all monoclonal antibody, as we would just need to change the sequence in the software, and the rest of the process would be the same. So the workflow is automated for the determination of the critical quality attribute at the subunit level. And we have SST on the LC and on the MS. So we believe it's applicable in a QC environment. The only thing which is not automated in this first part is the sample preparation. So this is how this can be solved by using a 2D system. So I will introduce here the 2D LCMS system we have at Quality Assistance. It's different modules from Waters with the Xivo G2XS, and it's also controlled by Unify in GMP compliance. And we have for this the multiple hard cut mode and the add column dilution technology. So I will explain this in more details in the coming slides. So let's discuss how the multiple hard cut to the LC works in practice. So this is an example of a first dimension chromatogram. And in this example, we are interested in 2 hard cut, 2 fractions from this chromatogram, from this elution that we want to analyze in the second dimension. So this is, again, the complete setup. So we have 3 pump systems, a QSM for the dimension 1, a BSM for the dimension 2, and another BSM called at-column dilution, which is used for the trap columns. So here is the configuration in the first dimension, where you see the blue line is where the flow goes, for showing the flow, how it goes through the different tubing of the equipment. So here in this setup, the flow goes from the QSM to the sample manager for injection of the compounds, all the way to the first dimension column, and is sent to the first dimension detector, the PDA in our case. So sorry, I said that there are 3 pumps, but there is also, of course, the sample manager, 2 column manager, 1 to feed the 2 columns from the first and second dimension, and 1 to feed the trap columns. And the QTOF is our second dimension detector. So this is what happens in the first dimension. And once you have here on the right top, the small scheme, once we reach the fraction of interest, we switch the position of one of the valves, and the flow is still coming from the QSM towards the first dimension, but the output of the first dimension, the compound, is directed to the trap column. And in addition to this, while we trap the compound of interest, we use the BSM called at-column dilution, which sends an additional flow, which allows for dilution of the mobile phase from the first dimension when there is too much salt, for instance. We also use this configuration when we want to do online DTT reduction, which I will explain also a bit later. So this is the configuration while we hard cut the first fraction of interest, and the compound remains focused at the entrance of the trap column, which is usually a small RP column. At the end of the elution of this peak, we go back to the first dimension configuration with compounds going to the PDA detection. Then we reach the second hard cut, and we have the same valve switch. We also can trap the second compound of interest, but we have to take care here. We have to use a second trap column, and in our configuration we can use up to 7 trap columns. So with one first dimension run, we can directly have 7 fractions of interest that can be analyzed on the second dimension. At the end of the gradient of the first dimension, there is the possibility to desalt further the compounds which are at the entrance of the different trap columns. So this would be the flow. Then we use the BSM at column dilution to send the flow to the trap column. And then the samples are analyzed on the second dimension. So we use the dimension 2 BSM. We send the flow from this column, backflush from this BSM, backflush the trap column, and then the compounds are eluted on the second dimension column and analyzed with the QTOF on the second dimension detector. If we have several trap columns with several compounds trapped, we need to run this second dimension gradient several times, once for each trap column. Now I will show you how we set up the disulfide bridge reduction online, so on this 2D system. We did the first test with TSEP that we solubilized in 10% acetonitrile. The antibody was, so we used Humira as a model compound for this test. We injected it on the trap column and started to send the flow of TSEP on this trap column to be able to reduce while it's still trapped on the column, and eluted on the second dimension with a bioresolve column with regular RP conditions. We tried the reduction step at different temperature. You can see the pictures on the right. We see the light chain and the heavy chain, but every time we see multiple peaks for both the light chain and heavy chain. And if we look at the MS data for this, we see different charge envelopes, and ones on the left side with the raw data. And if we deconvolute this, we see that there are shifts of 2 Dalton on each figures, which means that the intramolecular disulfide bridges were only partially reduced. So that's why we have several peaks. We have a mix of light chain with more or less a disulfide bridge remaining. So this was not satisfactory. This means the reduction was incomplete. So we tried to optimize this by increasing the time and the concentration of TSEP, but still this was not successful. We always had several species. So we decided to move on to DTT. DTT was not our first choice because there are more constraints on the pH. It requires a certain pH to be active, but we decided to dissolve it in ammonium bicarbonate to solve this pH issue. And we used the same setup as before, injecting the antibody on the trap column, sending a flow of DTT through the trap column, and elution on dimension 2 column. And here, what the chromatogram looks like with the elution of the light chain and the heavy chain. And in this case, we only have one peak of each, and it's confirmed by MS data that the reduction is successful. And we have also reduced all the intramolecular bridges as well as the intermolecular. So we decided to work with DTT. One other point of attention when working with DTT is the stability. So we compare the reduction with a fresh DTT and a DTT which would have spent 24 hours on the LC system, and the results were the same. You can see the chromatograms here. So we think that the solution can be used for overnight runs without losing activity. So all this leads me to the automated workflow for MAM analysis at the subunit level. To do that, we've again used the fabricator enzyme, but this time we have used it as an HPLC column. So the enzyme, the fabricator, is immobilized onto an HPLC column, which we can fit on the first dimension of our system. And the sample is directly injected on the column, and while it's going through the column, during the elution, the compounds are digested as expected as a IDS digestion. So for elution, we first tested PBS1X as eluent. This was only partially successful. It was not okay for all the antibodies we tested, so we decided, based on papers and discussion with Genovis, to use 200 millimolar ammonium acetate. And at the output of such a column, of the fabricator HPLC column, we see elution of protein based on the absorbance at 280, but there is no direct way to say that if the digestion was successful or not. So this is where we need the second dimension. So this is the complete setup. The dimension 1 is the fabricator HPLC column from Genovis with the elution with ammonium acetate. And we have to send a flow really slowly. So we use 0.025 milliliter per minute for 3 column volume. Then we have a trap column, which is again the bioresolve. And we use the map back column on second dimension with reverse phase conditions. And here we see on the tick, the chromatogram on the right, we see the FC peak and the FAB, which are the two expected species once you hydrolyze with fabricator. And this we confirm by looking at the MS. So the FC, which is 25 kilodalton, so on the left side, the raw data, the raw MS spectrum, and on the right side, the deconvoluted one. And for the FAB, we end up with a 97 kilodalton, which is also the expected mass. So the digestion was successful using this immobilized column. So we decided to continue and use this column on the first dimension, use the DTT reduction, disulfide bond reduction we discussed just before on the trap column, and elution on dimension 2 with a map back column. So these are all the steps we've seen before. So with the fabricator, immobilized, and the reduction. And we have the 3 species we expect. So this is another example with the antibody nivolumab. And we see the FC, the LC, and the FT. And this is again confirmed by MS, so the raw data and the convoluted spectra on the right, where we see 25 kilodalton for the FC, 23 for the light chain, and finally 24 for the FD. So this is also successful. This is working quite nicely without barely any human intervention, let's say. So to test the system, we again did a small case study where we prepared an nivolumab at 2 different levels of oxidation. And these are the chromatograms. So we see again the 3 species, and we see for the FC more or less oxidation. So for the first level, there is a bit of oxidation. And when we increase the hydrogen peroxide, we see the formation of 1 or 2 oxidated species on the FC peak. So eluting here around 9-10 minutes. And these are the unified data, which can be directly used with a processing method with automated identification and determination of the relative proportion of oxidation for each of the subunits. And here on the right, you can see the direct calculation of the level of oxidation for the FC peak. So of course, here it's an example with 70% of oxidation, which is quite huge, but still it's completely automated and identified. This was described in an article in LCGC in December 2022. So if you are interested in more details, this what I just showed now is described there on the 2D LCMS. So to summarize, we have developed middle-up approach by 2D LCMS with a fully automated sample preparation with the help of a immobilized enzyme on the HPLC column, so the fabricator HPLC column. We have an automated process with a GMP-compliant unified software, and we also have the automated determination of the extent of modification for 1 or several critical quality attributes. So thank you for your attention. Thank you, Philippe, and thank you, Genovis, for the invitation to present today. And I also thank my colleague at Quality Assistance. Thank you.
Wonderful. Thank you very much, Claire, for a great presentation. It really highlights nice use of fabricator, particularly the HPLC column, for a nice alternative approach to MAM for product attribute monitoring. So we've got a couple of questions have come in already, so we can start on those questions. So just bear with me 1 second. So the first question we've got is surrounding the injection of NIST as the SST. So do you inject the NIST as the SST at the beginning and at the end of the analytical run? And if so, do you see an increase over time in things like oxidation, mainly because the analyte stability, does it oxidize over time as it's kind of sat in the autosampler?
Yeah, so we do have it at least twice during a run. 3 times, I think, depending on the sequence length would be a good number because we want to calculate a percentage and error. So if you have triplicate, that's good. But at the moment, our sequence were not long enough to really check for oxidation, and also have to admit that this is not one of our criteria. So if there would be oxidation on the FC, we would not necessarily identify it because the criteria on the 2 other subunits.
Okay. So another question got another one about oxidation. So did you also detect oxidation on the FD? Is this impacted by the temperature on the trap column?
On the trap column, it's difficult to differentiate this from the, I don't know, maybe I don't have all the elements to answer the question. We can only measure the oxidation and the different attributes after the 2nd dimension run. So I'm not really able to say what happens during the trapping.
And what about the oxidation on the FD? Is this something that you saw, or were you looking for it? And if you didn't see it, could you detect it in this method?
Oh, we would detect it. I did not process all the data myself, so I don't know if this was so obvious, but yes, this is something we can see.
Okay, thank you. I've got one more here. So any idea regarding the variable abundances for the G0F minus N? So I think they're the ones you saw. They were quite low abundant ones, but they had quite high error bars in them. So was this confirmed by a release glycan approach? And go on, I'll answer that one as a second part of that question.
I don't know if this applies to the mAb or to the other antibodies. We have data on the adalimumab is usually our model antibody, so we have different results, and it correlates quite well on the glycan level. Yes, when we do the glycan analysis and the other approaches. Yes.
Yeah, so that question was referring to the mAb.
Okay.
I do have another question that I've seen is so how would this fare with mutated hinge region antibodies? I guess I could take that one.
Yes.
So the fabricator as it is, so that doesn't particularly deal that well with antibodies that have got mutated hinge regions rather. It's very specific in the way it binds to the antibody and then will do the cleavage. So in terms of the specific approach, probably not that well, or if at all. And currently, the only product that we have, or the only IgG protease that we have available on an HPLC format for this type of automated approach would be the fabricator. But we're always keen to have discussions about the potential for some other products on there, so we can follow up with that later on. But at the moment, this wouldn't deal that well with hinge mutated antibodies, this approach. One question, why such a low flow rate for the fabricator HPLC column? Could it be increased? I guess I can take that one as well. I think the main reason for that would be, I will clarify this response, but I think we want to ensure that we get full digestion on the column. I think the problem is if we have the flow rate is too high, there's going to be too high a risk of under digesting or not digesting properly. So we really want to ensure that we get full digestion on the fabricator HPLC column before we transfer onto the reverse phase column for separation. I have a question about specifically MAM, and of course MAM has its challenges, particularly when we're going into QC. Are there any particular challenges associated with, say, a traditional peptide MAM that could be alleviated by this middle up approach? And this kind of follows up, we had a second part to a question which I did miss, which I apologize for, the question of what are the specific advantages of a mid-level MAM compared to, say, a bottom-up approach? So I guess they're kind of similar.
Yes. I think the middle up approach is faster to set up and to obtain results. So this is it's the main advantage to me, that you obtain results quite fast. The peptide mapping requires more sample preparation, with the advantage of seeing maybe a few more modifications on the peptide level. If you're looking for deamidation, for instance, then of course I would recommend to go on the peptide level, but the subunit level, you directly have your answers on glycan and oxidation without too much time spent on sample preparation.
So how long does the digestion take on the I assume that's the fabricator HPLC digestion. So what for you, Claire, what was the digestion time?
Yes, so this is 3 column volume, so this is roughly 20 minutes. So this is just one LC run, which we also, this is maybe that's not that clear, but we can set all the method in our Unify software. So we have it goes directly from the first dimension. So this 20 minutes elution to the trap column to the elution on the second dimension is just one sample set. So we don't need to intervene here as well.
Thank you very much. Just scanning through to see if there's any more. So we do have a question actually about how does the Genovis fabricator compare to, say, other brands of IDES? Is that something that you've tried, Claire? Have you tried other brands of IDES and compared it to the Genovis brand?
No, I don't have experience on this myself. I've not tried any other brand.
Okay, so that's something we can follow up with offline for sure on that one.
Yes.
Good question to see if you tried any of the others. Where we can follow up on that one. I can't see any more questions that have come in. I'll give it another minute just in case any more have or do. I don't think I've missed any. But I can't see any others. Oh, hello. I think another one has just come in. So how robust are the Genovis fabricator HPLC columns? So I guess I will take that one. They are very robust. I think when we did the testing of them, we do have an application note actually when we developed this column from a few years ago, which is available for download from our website. And when we tested that, we did up to 14 days of consecutive running on this column, and it worked very well and very consistently over that time with minimal carryover as well. So in terms of the column, it's very robust and can certainly last a decent length of time, particularly for continuous running, which I think is very important for these kind of approaches, particularly if you want to do samples over a set period of time. So yeah, that's something. If you want further information, of course, we can provide that to you. And as I said, there's an application note specifically for the fabricator HPLC, which is available to download. I'm just seeing if there's anything else has come in. I think that's, oh no, another one more coming. That's good. So is Unify software, does Unify software process other raw files from other instruments such as Thermo or Bruker? Or is it have to be instruments?
Yes, this would be a question for Waters, but I think it's mainly Unify. It can, and you can have samples from other Waters equipment with data acquired on other software like MassLinks, but I don't know if it would work with Thermo and Bruker. This would need to be checked by Waters.
Yeah, I'm trying to think as well. I'm
not sure.
Since I've used Unify software, definitely that's one to check with Waters. Can the column be used in Shimadzu HPLC systems? I don't see why not.
Yes.
Yeah, it's just a column that can be, it's I think pretty standard in terms of how it is. It's just an HPLC column that can be plugged into any system. I would certainly say so. I would say that's not an issue at all. I guess what you'd need to do is make sure you had the setup to, if you wanted to do the automated approach, you need all the correct valves and everything and the pumps to make sure that you could do the 2D or the multi-dimensional approach. I think that would be fine. Any plans for a column version of Genghis Khan? We can follow up with that, Mark. Personally, I don't know at the moment, but we can follow up on that one for sure. So yeah, I'll be in contact with you. I'm not sure. To my knowledge, I'm not sure if it's something we've done before, but I can certainly check internally and get back to you on that one. So I think that is it. Oh no, there we go. Every time I say that's it, more questions come in, which is good. For the middle up approach, is it possible to monitor PTMs with small mass shifts like deamidation and isomerization?
Yeah, this might be where the limit for such method is, because it's difficult to see a 1 Dalton shift on a 25 kilodalton molecule. So it's not impossible, but this is not the method to use for such search. I would go for peptide level for this, yes.
I guess it depends if there's a capacity to change the second dimension to maybe say an ion exchange method, which could be something interesting, because then you might get separation of those, say the amidation certainly. But I guess using the setup that you've used with the reverse phase.
Yes, with the reverse phase, yes.
So thanks for that, Claire. I will again say there are no more questions currently. It's good. We've had lots of questions. I'm glad for that. So maybe we have another one. We do. So would MS2 be helpful? So would MSMS be helpful is the question. I don't fully understand. Would you be able to clarify what you mean by that, sorry?
If it's for deamidation, yeah.
Yeah, possibly. So I guess that would more, yeah. So I guess that for the amidation PTMs, go on, Claire, I'll let you answer before.
Yes, I think one delta shift, you don't have MS data for such MS2 data for such high masses, so this would not be helpful. You just need to see the mass on the deconvoluted spectra.
Yeah, and also then you're moving into more like kind of middle down approaches, which I guess would be a, from a QC perspective, that would be something that maybe would be, not particularly helpful. So I guess one question for me, I guess if we look into possibly, oh no, we got another one before I ask my question. So has quality assistance looked at native MS, such as ion exchange or SEC, with the online fabricator? So that kind of feeds into what we were saying before about changing the second dimension. Is that something you guys have tried?
We have tried ion exchange native, but not in the configuration. So that's a nice idea. We could fit MS-based condition on the ion exchange on second dimension. That would give probably other information and maybe a bit on deamidation indeed.
Cool. Thank you very much. So I guess that MAM and QC and MS and QC it's a very, it's been a hot topic for a while. So what would you say is, if you could put your finger on one of the most important factors of what it would, map to have a MAM approach in QC, what factors are kind of most important? Is it kind of the instrumentation? Is it the sample prep? Is it what kind of, what's important in that sense for you?
I don't know what you mean by important, like the variation,
I guess so. What would kind of what's kind of stopping it at the moment? Almost kind of like, why is it not more widely spread?
Well,
in a one line, one sentence answer. To put you on the spot.
Why is it not spread? Yeah, this is. I think it should, because it's really efficient, but it needs time to be accepted. It's not one day it's not, and the second day it's accepted. It's a long process, so I think it should be implemented in parallel to what is currently done. So we see also the relevance of such a fast and efficient method in parallel to what is currently done and people are comfortable with, for QC labs and for authorities, to show that this would work.
Yeah, thank you very much. One more question, not specific about the web. So would the recording be available? So yes, the recording of this will be available in the chat. There'll be, after the session is closed, there'll be a recording to be able to download in the chat. And I believe that we will follow up with everyone who attended as well. And in that, we'll have a link to the recording as well, so you can watch it all again, which will be very exciting for everybody, because I really enjoyed it. So I think now we can wrap up. I think we've had a lot of questions. And so, yeah, on behalf of everyone at Genovis, I want to say thanks again to Claire for coming and presenting for us today. It was really informative. And a huge thank you to everybody who else, all the attendees, everyone who showed up and gave us some of your time today. So thank you very much for that. I hope you enjoyed the session, and we hope to see you at more of these webinars in the future. We've done 2 now, and there's certainly going to be more to come. So please keep an eye out for details of upcoming Genovis webinars. As I said, once again, the recording will be available in the chat, so feel free to watch it again. And if anyone has any more follow-up questions about anything, feel free to drop us an email at any time, and we can certainly answer your questions and clarify any